The body can't just be making proteins constantly, otherwise we would all look like this. Hey guys, Fitness Science, and we're going to be speaking about the science of muscle growth. Part 1 of this video is going to be the biology, and then part 2 is going to be how you actually take this information and implement it into your diet in order to maximize your muscle growth in the gym. So from a cellular level, our body has DNA, which is found in the nuclei of our muscle cells, and DNA is basically sugar with a phosphate group and a nitrogen base of either A, C, G, or T, which are just the letters that denote which nitrogen base is attached to the DNA. But our body needs a way to make muscle protein synthesis with the information in our DNA, but it doesn't really want to take the DNA and use that because that's the original and we need to protect that. It would be like a builder going to build with the original blueprint when it's a lot more safer and efficient to take a photocopy of the blueprint and use the photocopies, because if the original blueprint is damaged or lost in any way, that's not really going to be ideal. And the equivalent photocopy in our body is called RNA, which is very similar to DNA, but it has a few key differences. It has different bases, so instead of ACGT it has AUCG, and basically what it does, it allows the body to read the instructions from the DNA without touching the DNA or risking damage to it at all. So in muscle protein synthesis there are two parts. There's translation and transcription. For transcription, to begin with, this is where parts of the DNA are used in order to guide the synthesis of the photocopy, which is RNA. So this is like taking a photocopy of the manuscript, and then the RNA will then go on to provide the instructions for the muscle building. So basically what happens, an enzyme called RNA polymerase attaches to the DNA, and out of all the free bases, the nitrogen bases that are floating around in the nucleus, it pretty much creates a long spaghetti of information that is a complete photocopy of the DNA, but it allows it to read the DNA, create what it needs, and then not touch the DNA anymore in order to risk damage. So the DNA never leaves the nucleus, but the RNA will, to provide the information for muscle building. So all the free bases attached to this long spaghetti strand of new mRNA, and this is the new photocopy on how to code for new muscle fiber. And the second part is translation, where the newly made mRNA moves out of the nucleus. It's got all the information it needs now from the DNA. It moves out into the cytoplasm, and these big subunits called ribosomes attach to it, and the ribosome pretty much pulls it through like a long spaghetti and reads the code as it goes through. Now the big ribosome is pulling this spaghetti through of mRNA, and it is reading the little letters 3 at a time. So it goes 333, it reads all the letters 3 at a time. Now what happens is the amino acids that we have in our body from our diet pretty much form the building blocks of the new protein that's being synthesized. And each amino acid has 3 letters that correspond to which amino acid it actually is. So for example, if the ribosome reads UUA, it will tell a little other molecule called tRNA to go and hunt out that amino acid and bring it back to the ribosome in order to attach it to the ever-growing and longer-growing chain of new protein. And that's essentially all our muscle is. It's a long chain of amino acids that is just attached to each other, spat out of the end of the ribosome and sent out in order to be laid down. But looking at the mechanics of the ribosome itself, it consists of 2 small subunits. The smaller subunit, which is called the 40S subunit, essentially sends out the tRNA and brings it back in and essentially starts growing the chain. But the bigger subunit, which is the 60S subunit, essentially provides the peptide bonds between the amino acids, or if you want to think about it like the glue between the amino acids in order to produce the new protein. Now, when our bodies are not in an anabolic state, i.e. we're in a catabolic state, either through fasting or we're not training or we're not having enough protein, essentially in this state, the 2 subunits are completely separated. They only come together when the body says, you need to make a protein, so come together and start working. And that's when, like I said before, the mRNA is pulled through the ribosome like a big spaghetti scanner. The sequence is translated into an amino acid, which the tRNA goes and gets and brings back to form the building block. And when the stop codon is reached, it will stop production because the protein is formed. And then the protein's released and it is free to be able to lay down new muscle. And then the protein will go and essentially form the basis of a new muscle fiber. It might be part of a muscle fiber, or it might be, a specific little part of a tendon or something like that. It will go and essentially be used for that. But the subunit of the ribosome called 40S, the smaller one, it doesn't just know where to go to find an mRNA molecule to attach to. It needs to be guided there. And the thing that guides it there is another molecule of an initiation factor called EIF4E. This is essentially the guide dog, which leads feeds the ribosome onto the mRNA spaghetti strand in order to start making a protein. But there is a way to inhibit this molecule from guiding the subunit to the mRNA, and this is essentially the way that the body turns translation on or off, because the body can't just be making proteins constantly, otherwise we would all look like this. The body needs a way to be able to turn it on and off, and when it's on, we'll be in states of anabolism, like when we've just trained, or we've eaten protein after a heavy resistance training session, and it will be off in periods of catabolism, like if we've had a fast for a day, or we haven't trained for a month, or we just are not producing muscle protein synthesis because we don't need it. And there are essentially two ways that we can turn this muscle protein synthesis on and activate this initiation factor to start binding the ribosomes onto the mRNA in order to then start pulling it through and starting to create protein. And the two ways in which we do this are through diet and through training. Diet I'm going to speak about in part 2 of this video, but with the training, essentially our body has a number of mechanotransducers. When we subject it to resistance training, these are stretched and these open up, which through a complex pathway of reactions allows for the synthesis of a certain acid, which then activates mTOR, which is a huge pathway which guides muscle protein synthesis. That then activates the initiation factor, which we spoke about, EIF4E, which then can guide the ribosome onto the mRNA, and it can begin its work in producing a complete new protein. This essentially is how our body increases the rate of muscle protein synthesis. So resistance training does increase the rate of protein synthesis through the mechanotransducers being stretched open and essentially producing an acid, which then goes downstream and turns on mTOR. And the second part is going to be on a specific amino acid, which we actually take in through our diet, which has been shown to increase and stimulate mTOR and therefore muscle protein synthesis to a really large degree. And it's one of the only amino acids that can actually do this. And that's why in part 2 of this video, I'm going to speak about how you actually optimize your diet to increase the ingestion of this amino acid. Thank you so much for watching the part 1, which is the biology of this video. It was a bit complicated, but I hope that some of these points make sense, especially around why your body will actually go through muscle protein synthesis when you resistance train. And then diet is coming in part 2. I really appreciate all the support, and I'll see you guys in the next video.